Ligation of the B-cell receptor and CD40, together with direct contact with T cells, are all required to sustain germinal center B cells. Germinal center B cells are inherently prone to die, and in order to survive, they must receive specific signals. It was originally discovered in vitro that germinal center B cells could be kept alive by simultaneously cross-linking their B cell receptors and ligating their cell surface CD40. In vivo, these signals are delivered by antigen and T cells respectively. Additional signals are also required for survival, which are delivered by direct contact with T cells. The nature of these signals is still obscure, but one signaling system involving the TNF family member BLYS, the T cell signal, and TACI, its receptor on B cells, has recently been found to be essential for the maintenance of germinal centers. The source of antigen in the germinal center has been the matter of some controversy. Antigen can be trapped and stored for long periods of time in the form of immune complexes on follicular dendritic cells, and it was therefore assumed that this was the antigen that sustained germinal center B cell proliferation. While this may be true under certain circumstances, there is now evidence that antigen on follicular dendritic cells is not required to sustain a normal germinal center response. Indeed, the role of the antigen depot on these cells is unknown, although it could be to maintain long-lived plasma cells. Where does the antigen that sustains the germinal center come from? Under normal circumstances, it is most likely that live pathogens carried to the lymphoid tissues and multiplying there will continue to provide antigens until they are eliminated by the immune response, after which the germinal center decays. Immunizations with protein antigens are usually given in a form that slowly releases the antigen over time, which mimics the situation with live pathogens. Indeed, it is difficult to stimulate germinal center formation by immunization without either a live replicating pathogen or a sustained release of antigen in adjuvant. How the various signals that maintain the germinal center exert their effects on B cells is not completely understood. The combined signals from the B cell receptor and CD40 seem to upregulate a protein called BCL-XL, a relative of BCL2, which promotes B cell survival. There are doubtless many other signals yet to be discovered that promote B cell differentiation. And that's ligation of the B cell receptor and CD40, together with direct contact with T cells, are all required to sustain germinal center B cells. Have a great day.